Okay, so this is RNA transcription. Right, so RNA transcription happens in 3 steps. Initiation, elongation, and termination. So we have here the DNA antisense, which will be transcribed, but the sense strand is the gene that we want. So we're going to start with RNA polymerase, which does all the work. The RNA polymerase will find the Tata box, or the promoter gene, where it begins building the DNA from a 5' to 3' side. So starting at the Tata box, we're going to build the RNA strand. The difference between DNA and RNA is the uracil. So the T is instead of A and T, it's A and U. So we're going to build along the antisense strand. We're going to build DNA all the way down. Oh man, this is rough. U, G, A, G, C, U, and as we're going along, the RNA polymerase is taking the RNA bases and putting it down in against the DNA bases. And this process is elongation. We already passed termination, or we're already past initiation, because initiation was the RNA polymerase finding the promoter gene. Alright, and it goes all the way down.
So just
G, A, C, C, A, T, or A, and C. So once the RNA polymerase builds the RNA gene all the way down, and it sees the gene that codes for ATC, it knows when to stop, this is the terminating gene. So this is where we reach termination. the sequence of termination. Now the elongation is over, the RNA is set, it's now premature mRNA. And so it separates from the DNA goes back to normal, and it moves on. Alright, and so the RNA separates from the DNA as it goes. Alright, so DNA removes itself, it forms that hydrogen bond back together, and it moves on. While the mRNA that we have, it can't leave the nucleus just yet because it still has a lot of unnecessary genes that doesn't really code for the protein that we need, which are called introns. And so the introns we have are from the 23rd base to the 33rd base. So we're going to take those bases out. Where is it? Out, and then this is called DNA splice or RNA splicing. So these introns are out, which what we are left with are all the exons. So the exons connect with each other again, creating mature mRNA. But still, at this point, it can't leave the nucleus yet because it doesn't have any coding that tells it it can leave. So what we need to add to it is on the 5', we need to add the G base, so that it's able to leave the nucleus, as well as the poly A tail, so that it can be protected while it's in the cytoplasm traveling to the ribosome. So we have a bunch of poly A tails on this end, so it can leave the nucleus. So right now, this mRNA is mature enough to leave the nucleus, and so we have done initiation, elongation, termination, and we have done RNA processing, which is adding on the 5-methyl cap and the poly A tail, so that the RNA can leave the nucleus. All right.